Hello and welcome to Chapter 3. We're going to talk about cells, a little bit about the variation in cells, and then we're going to get look into the process of transport in cells. So when we look at cells, there are great variety in cells. And if you look here, this is a nerve, and nerves can actually be very long. If you consider that the nerve that goes all the way to your toe would start in your lower back. So that's almost a meter long. And then you've got muscle cells, which are quite detailed. Blood cells, which don't have nuclei in them, but they have an interesting developmental process. And then you have gland cells. So you have different cells. And we'll look at the different cells when we look at specifically at the different units that have these cells. Now, there is this generic cell structure. And the generic cell structure has all the parts of the cells. Now, being a little cell, it's called an organelle. The easiest way to look at it is to start with the main center in the middle. Now, when it comes to organelles, you need to know the name of the organelle and the function. So let's start at the beginning, in the middle. In the middle of the cell is the nucleus. And the nucleus has its special membrane, which has different holes or pores that allows things to go through. The nucleus has a center that has concentrated genetic material, and this is called a nucleolus. Inside the nucleus is where you find the DNA or the chromosomes or the chromatids. And this is the site where there's organization. This is the part that commands the activities of the cell, because the DNA will code for the proteins, which then code for the function of the cell. Outside the nucleus are these flattened sacs. Now, there's two types of flattened sacs. There's flattened sacs that have little dots on them, and there's flattened sacs without dots on them. And then there are flattened sacs, but there's a third type that isn't connected to the nucleus. So the flattened sacs connected to the nucleus are called the endoplasmic reticulum. Endo means in, reticulum means straight. So they're the straight sacs within the cell, within the plasma. Now, the nucleus will give the information on how to code a protein. It will then come up as RNA is sort of the copy, messenger RNA. The messenger RNA will land on the ribosomes, which are these little dots. And the ribosomes work as work benches. Let's try that again. Work benches where the genetic material is matched up. So the messenger RNA codes are matched with the transfer RNA, which will then bring an amino acid, which will join together to form a protein. And all this happens on the ribosome. So the ribosome is the site where it happens. And then after these proteins are made, they are put into the endoplasmic reticulum. And in the endoplasmic reticulum, that's where they are folded and modified. Now, in the smooth endoplasmic reticulum, that is where the lipids are created, folded, and modified. Then they will branch off. The endoplasmic reticulum branches off these little pods called vesicles. These vesicles will then join together to create what is called the Golgi apparatus. Now notice Golgi is a capital G because it's named after a guy named Golgi. And the Golgi apparatus is where there's further modification. So the lipids and the proteins are put together and they're modified with more detail. And this is where sometimes it's called the post office, because they get special processes on them, which will then tell them where to go. I have a video that explains a little bit about the protein trafficking, or how a protein moves through the cell. Now, it's very technical in terms of a lot of words, so I'm actually going to shut the volume off and we'll just skip through it. But I think it's really good at showing what's going on. So When we talk about the Golgi apparatus receive proteins from a location and target them to send into a second location. So they are often called the post office. Now, when you look at Golgi, there we go. The Golgi apparatus consists of these components. A lot of terminology, but here's the ER. So the endoplasmic reticulum will send off these little packages, which will then fuse together. And you'll have these stacked type of pancakes, which will then process things. So the proteins are transferred. And there's many enzymes in these situations. But I want you to pay attention how here are the vesicles that come off of the endoplasmic reticulum. Now, because of these dots, this is rough endoplasmic reticulum. Rough endoplasmic reticulum will make proteins. So the proteins are coming off, and they will join together to create a layer of a Golgi apparatus. Then, as they create this layer, inside there are enzymes, and these enzymes will then modify the protein. And as they modify the protein, it determines the destination. Then the neat thing about it is these stacks will move through the cell with new ones coming, forming new layers. And then when they get to the end, the Golgi apparatus will make vesicles, which will then go to the outside of the cell. And so here you see that it just shows the color changes, shows enzymes being modified, enzymes doing the modifying, and then they will produce these vesicles, which will bud off. And then once they are done budding off, these are, depending on what's in them, they can have chemicals for digestion, such as lysosomes. They can have sacs that need to go to other part of the organism. So they will then fuse with the cell membrane. And once they fuse with the cell membrane, they will then go and deliver the stuff to the outside. So here you can see the vesicles from the endoplasmic reticulum form the Golgi apparatus. The Golgi apparatus will then send off vesicles. These vesicles will then fuse with the plasma membrane to put the nutrients outside. Now, the plasma membrane is the boundary of the cell. And we talked briefly about how the plasma membrane is made of this phospholipid bilayer, which is two layers of these molecules that have a polar end and a nonpolar end. And so it provides for this movable but yet strong structure on the outside of the cell. Okay? So we've talked about the nucleus in the center, the endoplasmic reticulum. We've talked about the Golgi. Now, you need energy in order to have the cell work. And the energy is created in a organelle called the mitochondria. The mitochondria is folded inside. There's enzymes on these layers, and this is the mighty mitochondria, which is the powerhouse of the cell. And this is where the ATP, the glucose, is broken down into greater detail, producing carbon dioxide and water, and that molecule called ATP. Okay. The cell has structure to hold it together. There's plasma in the inside, and we talked about that being the intercellular fluid. And then inside you have got proteins and fibers. So microtubules, microfibers, that is the cytoskeleton of the cell. Then in an animal cell you have centrioles, and centrioles are used in the process of mitosis, which is the process where the cell divides. The centrioles act as poles at the end in which the spindle fibers are attached to, which help pull the chromosomes apart. Now, some cells have the ability to move, so they can have cilia on the outside. Cilia is a hair-like projection which will allow for movement. And this is found in your throat. It's found in the oviduct. Okay? Now, when we look at the membrane, we talked about the cell membrane having this specialized phospholipid layer. So there are two layers. There is a molecule inside. Now, this is cholesterol. Can you tell by its shape what class of molecules it is? See how it's got carbon rings? This is a steroid. So cholesterol is a steroid, and cholesterol, although people talk about being bad for your cells, cholesterol is the building block for a lot of hormones, especially estrogen and progesterone and testosterone. Now, on the outer side of the plasma membrane can be these carbohydrates, or sometimes called carbohydrates. And the carbohydrates will act as little antenna or receptors. And these receptors will be what signals other cells to come and land on them. Now, the membrane itself only allows small non-charged particles through. So there is a channel which allows other things to go through. And these channels are made of proteins. And they allow for things to pass. So let's look at the proteins. Proteins are embedded in various spots, and we talked about that when we talked about proteins. Most pass all the way through and act as channels. Some are on the surface and act as receptors. And most have carbohydrates attached on the outer surface, and those are glycoproteins, and these can be markers. When we talk about proteins and receptors, one of the things, if you've been following the production of the COVID vaccine, it talks about the spikes on the outside. The COVID virus has a spike on the outside, and that's what gives it the corona look or the crown look. And these spikes are proteins. So in order to have a vaccine against it, the mRNA or the vaccine gives you the instructions to produce these proteins. Okay? So proteins are markers on the cell. We talked about the nucleus, double layer. We talked about the nuclear pores, which allow things in and out. The nucleus the DNA never leaves the nucleus. It's messenger RNA that takes the information in and out. Then we talked about the ribosomes as the place for protein synthesis. So they're like the workbenches. We talked a little bit about the endoplasmic reticulum. The endoplasmic reticulum can be smooth and it can be rough. And in smooth, they have, smooth has the enzymes to make lipids and carbohydrates. Rough has the enzymes to make proteins. We talked about, in the video, we talked about how the proteins are made, and then the proteins are pinched off in a transport vesicle. These transport vesicles fuse to the Golgi apparatus. They get modified as they go through. They create whatever needs to be created, and it's deposited in the secretory vesicle. So the vesicles are transport pods. We talked about centrioles. Now, when you look at the mitochondria in more detail, you'll see it's a double-membraned organelle. And we talk a little bit more about this when we talk about energy creation, but there's the matrix, which is another word for space. And then you have the cristae, which are the folded fingers inside. The cell is given strength using what's called cytoskeleton. And the cytoskeleton is made of protein filaments, and these protein filaments are microtubules, microfibers, and intermediate filaments. The difference between them has to do with size and structure. We won't get into detail here, but that's what gives a structure in the cell for strength, but also for cells and molecules to move along them. And let me find that video for you. Okay, here's a video that shows how these are the microtubules, which are the part of the cytoskeleton, and this shows how it is a molecular framework in which molecules can walk on.
It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that very broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way. Exploring at the...
So those molecules with the little legs are a way of showing how these microtubules and microfilaments are used as pathways for molecules to move along. Now, we've talked about cilia. Cilia is like the hair-like structure that will move substances along. In your throat, it will move any sort of foreign particle back up to your mouth. In sperm, you have a moving, an organelle used for movement, but this is a flagella. Flagella is more whip-like. Cilia is more like wave-like or rolling. Okay, and that is the end of the talk on the organelles, or the parts of the cell. So I'll stop the video here and I'll make another one for transport.